Hello, welcome to Adaptive Immunity 5. So this is the final video for chapter 16. So I'm calling it a synthesis of everything we've learned about B cells and T cells and macrophages, natural killer cells, clonal deletion, clonal expansion, etc. Synthesis. Okay, so this slide is entitled Inducement of T independent and T-dependent humoral immunity. So we mentioned this in the last video for chapter 16, but in this cartoon it says T-independent humoral immunity. So there's some clues there. When you see humoral immunity, you should automatically think B cell. And then when you think B cell, you should think plasma cell. You think plasma cell, you should think antibodies. But in this cartoon, it shows our hero, B cell, and then these Y things sticking out of its cell membrane. Those are B cell receptors. And what it's doing is the B cell receptors are recognizing this green thing, which they're calling a polysaccharide, or many sugars, with repeating subunits. So in this cartoon, the B cell recognizes this polysaccharide, and it doesn't need a helper T cell to activate it to become a plasma cell. So B cell recognizes this polysaccharide. Without the help of a helper T cell or any cytokines, it differentiates into plasma cells, and then makes antibodies, which are pretty much identical to the B cell receptor. And then these antibodies will go out looking for more of these polysaccharide repeating subunits. So that's T-independent humoral immunity. And then this is a cartoon of T-dependent humoral immunity. So here's our hero, the B cell, and here's the Y sticking out of its cell membrane, which are the B cell receptors, and then these little green dots are the antigens. So the B cell recognizes the antigen epitope, and then so then you come down here with the help of a helper T cell secreting cytokines. So in this cartoon, they're calling the cytokine helper factor, but really it's a cytokine. The helper T cell will help the B cell become fully activated, and what that means is the B cell then forms a clone, and then those clones form plasma cells and memory cells, and the plasma cells secrete antibodies into the blood, and then the antibodies will recognize these little antigens up here, and only those antigens / epitopes. And all this is happening in a lymph node, specifically the germinal center, so where B cells and T cells are becoming fully activated. How do you induce T-independent humoral immunity? So you can use these large antigens. So they called it a polysaccharide in the last cartoon, but here we're calling it just a large antigen with repeating identical epitopes. So each one of these little green balls is a repeating identical epitope, and it's recognized by these B cell receptors on the B cell. If you are a large antigen with repeating identical epitopes, you can induce the B cell to make plasma cells and then antibodies without the assistance of a helper T cell. That's T-independent humoral immunity. Most B cells, however, cannot be activated without assistance from T helper cells. Most B cells need a helper T. Some T-independent antigens, so B cell receptors will recognize molecules on capsules. They'll recognize LPS. Remember, that's in gram-negative bacteria. B cells will recognize bacterial flagella, capsids of viruses, and then they'll all, once the B cell recognizes all these things, they can go on to differentiate into plasma cells and make antibodies without the help of a T helper cell. Okay, still more on T-independent humoral immunity. When a B cell engulfs a specific pathogen that it matches, so the B cell receptor recognizes the pathogen, the specific pathogen, it will become slightly activated. So it's activated enough to make plasma cells and antibodies. You can also call this process T-independent activation. So these B cells are getting activated to form plasma cells without the help of a helper T cell. That's T, you can call that T-independent activation. So T-independent responses are really fast. You don't depend on T cells. It gives you weak immunity, however, so the antibodies disappear quickly. And then it just gives you some factoids down here. T-independent responses are stunted in children, so they don't have their immune system fully developed. And then it tells you Haemophilus influenzae capsules, so around that bacteria, do not stimulate B cell dependent immunity in children, and that results in most cases of meningitis in unvaccinated children, but the adults are not. Now we'll talk about T-dependent humoral immunity with clonal selection. So T-dependent immunity begins with the action of an antigen-presenting cell. So here's our antigen-presenting cell. Remember, antigen-presenting cells have MHC2 proteins in their cell membranes. Usually the antigen-presenting cell is a dendritic cell. So the antigen-presenting cell, it doesn't show it in this cartoon, but previous to this cartoon, or this step in this cartoon, the antigen-presenting cell had a bacteria or a virus on the inside. And remember, it chopped it up, and then it took a piece of the pathogen, which would be this green diamond, and it put that epitope from the pathogen into the MHC2 complex here, and then presented that on its cell membrane, and then a T helper cell with its T cell receptor specifically found this epitope MHC2 complex, and then you form this immunological synapse. And all these cluster of differentiation proteins, or CD proteins, they all are just kind of co-receptors to help stabilize the synapse and make sure that the recognition is valid. Okay, so antigen presenting cell presents antigen epitope in its MHC2 complex to a helper T cell with a complementary T cell receptor. So that word complementary just means that the T cell receptor recognizes or is a complement to the epitope MHC2 complex. Then the activated helper T cell becomes a Th2 cell, and then that T helper cell can activate a B cell that has also recognized the same antigen with its B cell receptors. So think about that. That's 3 cells now. That's an antigen presenting cell that initially found the pathogen with its epitope. Recognition by the helper T cell of that epitope on the antigen presenting cell. And then there's also a B cell that it's not shown in this cartoon. I think I showed it in the next cartoon. But the B cell will also recognize that same antigen. And then there'll be some cytokines secreted between the helper T cell and the B cell. B cell will make plasma cells. Plasma cells make antibodies. Antibodies will travel all over the body and destroy this initial guy right here, because those antibodies will recognize this piece, the green diamond, on those bacteria. Oh, here's the cartoon of the helper Th2, helper T cell number 2, with its T cell receptor can recognize the B cell presenting that same epitope, this green diamond, that same epitope that was in the first cartoon. So then you've got the helper T cells, the antigen presenting cells, and the B cells all working together to activate the B cells so that it makes antibodies that will kill that original pathogen. And this is all happening in the lymph nodes. Okay, so this slide's called T-dependent humoral or B cell immunity with clonal selection and clonal expansion of plasma cells. So we kind of looked at this cartoon before. This is a little bit more detail. It says immunocompetent B cells. That means that the B cells have matured in the red bone marrow and now they're in the lymph node and they are competent to do their job, which is to recognize antigen epitopes with their B cell receptors, their complementary B cell receptors. So then it shows here that the B cell displays processed antigen fragments. So it doesn't really show that very well, but the helper T cell will bind to those antigen epitope fragments and secrete helper factor, which you can call them interleukins or cytokines, and then that stimulates the B cell to divide and form a clone. So that's where we get clonal expansion in the title of this slide. Clonal selection is just when this original B cell gets selected to form a clone because this original B cell bound to that specific antigen. Some members of the clone become memory cells, most differentiate into plasma cells, and they make antibodies. So let's look over here at our check mark. It says once activated by a helper T cell, which is going on right here, B lymphocytes undergo rapid cell division, mitosis, and that's the process of clonal expansion. And then during clonal expansion, B cells immediately, plasma B cells. So some people call them plasma cells, people call them plasma B cells because they used to be B cells, but now they're plasma cells. So in this check mark, we're calling them plasma B cells, are immediately active. That means they can make antibodies. And the memory cells are immediately inactive. So the memory cells won't come into play unless that same pathogen with this antigen comes back and infects your body. Okay, more cartoons on T-dependent humoral immunity with clonal selection. So remember, this is all in a lymph node. So here's some helper T cells, aka CD4 positive cells, with their T cell receptors, and then an antigen presenting cell. So this just the shape of this cartoon cell looks like it's a dendritic cell. So this dendritic cell probably was in the skin, phagocytized a pathogen, chopped it up, and put a piece or an epitope of that pathogen in this MHC2 complex, and this T cell specifically had the receptor that recognized this. So here's the helper T cell. This specific T cell receptor recognized this epitope in the MHC2 complex. These guys had T cell receptors, but they did not recognize, because remember, this is a specific event, specific immunity. So this helper T cell specifically recognized that antigen presented by an antigen presenting cell. And then this cell, this T cell, formed a clone. So now you've got a clone of these T cells with this specific T cell receptor on them. And then it shows there's some IL-4 secreted, so that's a molecule that will help the helper T cell differentiate into a helper T cell number 2. Okay, and now you have, and then over here, so this is all in the lymph node, remember, so you've got B cells and T cells and antigen presenting cells in the lymph node. You've got this repertoire of B cells, and here's MHC proteins in their cell membrane, and this B cell will be clonally selected because it's found with its B cell, it's found this same pathogen with this same epitope. So at this point, the helper T cell will recognize the epitope. So remember, it's a green epitope here and one here, this diamond, green diamond. So the helper T cell will, helper T2 cell will recognize this epitope in the MHC2 complex in a B cell. And it looks here like the helper T cell is secreting some IL-4 that will help to fully activate the B cell to form a clone of plasma cells that make antibodies. And then that activated B cell will also form these memory B cells. It's all happening in a lymph node. All right, so let's break this down. Antigen presentation for a helper T cell, activation and cloning. So antigen presenting cells, so for example, our dendritic cell here, after acquiring antigens, moves to a lymph node. So LN is lymph node, takes about a day. So what it's not showing in this cartoon, what they mean by this is this dendritic cell was living in the skin. Toll-like receptors in the cell membrane recognized PAMPs on a microbe, for example, a bacteria, chopped it up, took it inside, the antigen presenting cell chopped it up, and put a piece of the antigen epitope from that bacteria in its MHC2 complex, and then went to a lymph node to find a T cell, a helper T cell, with a helper T cell receptor that could actually recognize specifically this epitope MHC2 complex. So helper T cells use their T cell receptors to survey all antigen presenting cells for complementary epitope MHC2 complex. So that means these T cells here, they took a shot at binding to this MHC2 epitope complex from the antigen presenting cell, but it didn't happen. They didn't hook up. Only this T cell was able to specifically recognize the epitope in the MHC2 complex. And when they bind, like in this cartoon, so here's the helper T, here's the antigen presenting cell, when this T cell receptor recognizes the epitope MHC, that they form an immunological synapse. And then the CD4 molecule on the helper T cell stabilizes it. And these other molecules are there too. And then it says the antigen presenting cell secretes a chemical signal that induces the specific helper T to proliferate, making a clone. So it doesn't really show it here, but this antigen presenting cell would secrete some cytokines that would bind to this T cell to help it along its path of differentiating into a clone. Okay. So they don't know, in this cartoon, they don't know where the IL-4 comes from, but the IL-4 signals differentiation of these helper T cells into helper T cells phase 2, or helper T2 cells. Okay, and then while that's going on, you've got the B cells over here. The B cell receptor attaches to and phagocytoses a complementary antigen. So just like with the dendritic cell, this B cell, they're not showing you what happened upstream of this cartoon. So not shown in the cartoon, you have a B cell. Its B cell receptor is a attaching to some specific antigen on a microbe, and then the B cell takes in that microbe with its antigen epitope and puts the antigen epitope in a specific MH2 protein on the surface. So it says antigen binding primed clonal selection of B cells. So this is important. This is just primed. So when the B cell receptor bound to the antigen, doesn't show a picture of it, that primed the B cell, getting it ready to form a clone of B cells. So B cells and helper T2 cells can now interact to fully activate the B cell. So yeah, here's our B cell. It's got its epitope from the pathogen, and then this T cell receptor will recognize that, and the helper T2 cell will secrete cytokines and fully activate the B cell. So the helper T2 cell binds to the B cell in this immunological synapse, and then B cells are triggered by a chemical signal, so IL-4, from the T helper 2 cell, and then that fully activates the B cell. It forms a clone in memory cells of B cells. This is just a helper T cell summary. So here's a macrophage, B cell, or some other antigen presenting cell, which could be another antigen presenting cell, could be a dendritic cell. So helper T cells are here. When helper T cells bind to the antigen presenting cell, they can play a role in non-specific defense, humoral immunity, and cellular immunity by secreting these cytokines. I think that might be it. Maybe not. Let's see. Oh, yeah, there's more. How do antibodies destroy pathogens? So we went over this when we did the B cells in the previous slide, but so here's a microbe, probably a bacillus. It's got some antigens sticking out of its cell membrane, and here's an antibody, and antibody binds to the epitope antigen on the microbe, and then that starts this complement cascade that leads to a lot of different pathways, including enhanced phagocytosis by macrophages, and also these complement proteins can punch a hole in the microbe. Antibodies also destroy pathogens via this process of neutralization. So here's a bunch of antibodies, these Y-shaped red things. They're binding to this green triangle toxin. Antibodies can bind to adhesin proteins on a bacteria, or these adhesin proteins, which are the spike proteins on a virus. And you can neutralize the toxin, bacterium, or virus this way. Antibodies can bind to the antigen epitopes on the bacteria, and then the antibody, the Fc region of the antibody, can bind to this Fc receptor, and it enhances phagocytosis. You could also use your antibodies to just agglutinate or clot all of your microbes together, and then they can be destroyed in the spleen, for example. And then natural killer lymphocytes. If antibodies are bound to an epitope on a microbe, again, the natural killer lymphocyte has this Fc receptor here that can bind to the Fc portion of the antibody, and then the natural born killer lymphocyte can put perforins to bust open the membrane, and then dump in granzymes, so kill the bacteria by apoptosis. So these are graphs of time periods in primary and secondary response to a pathogen. So if you look on the y-axis, it says the antibody concentration in the serum, and on the x-axis is the number of days. So in the primary response, that's the first time you see the microbe. The secondary response is the second time you see the microbe. So primary response, you have this lag period because it takes a little while, up to 3 days to start making antibodies, but you start making IgM first, then you make IgG, and then some cells will make IgA or IgE. These individual plasma cells that are making the antibodies for you, they're short-lived, but the antibodies can stay around for several weeks. You can get a secondary response to a pathogen if the memory B cells encounter the same antigen again. So here's exposure, for example, they use tetanus toxin. You start making, in the second time you are exposed to tetanus toxin, you start making IgG and IgM really quickly. And sometimes you don't even know you're sick. Okay. So this just says some of the induced B cells become memory cells. We've already talked about that. The memory cells can live a long time, maybe sometimes for years in lymphoid tissue. Let's take tetanus immunization as an example. A few helper Ts and B cells bind to the tetanus toxoid, and in the primary response, you'll make a small amount of antibodies. So yeah, check this out. In the first response, you make this much IgM. It only goes up to about here on the y-axis. In the secondary response, you make maybe about the same amount of IgM, but you make a lot of IgG really quickly. And then that antibody can bind to the toxin. If you get a third exposure, you get an even more effective response. So you start making IgG and IgM even faster and in greater amounts in the third exposure. So this is just a flow chart that kind of brings it all together. Here's on the left is humoral or antibody-mediated immune response. So that's B cells. Remember, humoral response is B cells and antibodies. And then here's the cell-mediated immune response. Those are T cells. So remember, if the pathogen gets inside the cell, infects the inside of the cell, then you use the cell-mediated response. But if the pathogen is out in the blood, the body humors, then you use the antibody-mediated immune response. And then it just it shows the first exposure of the antigen. Macrophage engulfs it, and macrophage, along with the helper T cell and the B cell, or the cytotoxic T cell. So the helper T cell helps the macrophage, the B cell, and the cytotoxic T cell. But then the B cells or T cells will form clones with memory cells, and then in the second exposure to the antigen, the immunological response is even more deadly to the pathogen. And this is just writing. You can read through this, just sort of a summary of everything we've been talking about.